At the production site, the drug is made into a herbal medicinal product.
EPS 7630, or to use the Latin term Pelargonium Sidoides extracts, are among the best studied phytopharmaceuticals we have today. As far as efficacy is concerned, the situation with regard to studies is excellent. To my knowledge, there are close to 10,000 patients involved in studies, and almost half of these in controlled trials. What's particularly important for this indication is that it has a lot to do with children. There are also studies available which involve children.
In order to get a marketing authorization for medicinal products, the first thing one has to do is ensure a consistently high level of pharmaceutical quality. With herbal medicinal products in particular, this is a major challenge because these products consist of a multitude of ingredients which quite obviously complement each other in their mechanisms of action. Thus, the total extract is typically more effective than the individual substances as such.
In herbal medicine products, we have not only one structure, but actually several fractions. And each of these has a specific pharmacological effect. Thus, we commonly use the term pleiotropy, that is from pleios, meaning multiple, and treptine, meaning to address. So, with all the different individual fractions, I'm addressing different receptors or different mechanisms, which means I'll get a completely different profile of effects. And this is a special characteristic of EPS 7630. It has a partly antiviral, antibacterial, and anti-inflammatory effect. It has a corresponding effect on the mechanisms which trigger or perpetuate a cold. And this is why phytotherapy is so fascinating, because these are multi-component mixtures with several individual fractions. So I've got a specific mechanism with each fraction and don't have to give 3, 4 or 5 different things in combination. That's the difference, and that's what makes natural medicine so fascinating.
This is why an accurate analysis of individual extract components is of essential importance, not only with regard to the mechanism of action, but also in terms of tolerability. With EPS 7630, this applies in particular to coumarin derivatives, of which it contains small amounts.
EPS 7630 contains 7-hydroxycoumarins, but not the coumarins we have in anticoagulants, such as fenprocumin or warfarin, for example. In their structure-effect relationships, they differ in 2 significant places, namely at the C3 position and at the C4 position. In one position, there is an OH group, in the substances with an anticoagulant effect, such as marcuma, for instance, and the side chain, which accounts for absorption. Typical of EPS 7630 is that it has no effect on coagulation. An interesting aspect is that coumarins are metabolized in completely different ways. Through the inoculation of amcalin, which is the primary substance in EPS 7630, with human liver cells, it was established that no toxic metabolites are formed in the degradation process.
The exact pharmacological profile of effects was investigated by scientists from the University of Freiburg in Germany. As is already apparent from their ongoing research, the anti-infective effects observed so far are directed against a broad spectrum of pathogens.
We did a total of 7 studies in order to find out what effects does this substance have. And one would have to say its effects are primarily immunological.
Thus, for instance, it has been proven that the risk of adhesion of viruses and bacteria was substantially reduced by a protective film coating on the mucous membrane cells. So, in a manner of speaking, their attack is thwarted right at the very surface. But even if, for lack of such prevention, the viruses have already gained ground on the mucous membranes, Pelargonium Sidoides is still effective. It activates the body's natural defense system. Natural killer cells are mobilized. They enter the mucous membrane cells that have been seized by viruses and kill them off. This deprives the virus of its basis for reproduction, while also leaving it susceptible to the body's defense system. This is where the macrophages come in, so-called scavenger cells, which are also powered to top performance by EPS 7630. Like the tentacles of an octopus, the evaginations of these cells move in on the intruders from all sides, leaving them no chance for survival. But the scavenger cells don't stop at that. They also ingest and destroy any other cells that are injured or damaged until the mucous membrane is thoroughly cleaned of any debris that interfered with its normal function and can recover. Bacteria don't stand much of a chance either when exposed to this natural extract. Its active substance renders them unable to multiply any further. Stimulated by Pelargonium Sidoides, the cilia of the mucous membrane simultaneously increase their frequency by up to 40%. Mucus is removed more efficiently, the nose begins to run, the swelling of the mucous membrane subsides, the airways open up, the infection is under control. For a medicinal product to obtain a marketing authorization in Germany, studies regarding its pharmaceutical quality and mechanisms of action must be complemented by trials with human patients to confirm its efficacy and tolerability. Thus, more than 3,800 patients were involved in controlled double-blind trials and more than 5,400 patients in open-label trials and post-marketing surveillance studies. Specifically, 3 placebo-controlled trials conducted in adults with acute bronchitis were instrumental in obtaining the marketing authorization. The assessment of efficacy was based on the severity score of typical bronchitis symptoms. Over the course of 1 week, the total score, which included such cardinal symptoms as cough, sputum production, rales, chest pain and dyspnea, decreased significantly faster with EPS 7630 than with placebo. This is an essential factor with regard to the patient's ability to return to their workplace sooner. The fact that the period of working inability is reduced by more than 2 days with EPS 7630 as compared to placebo can be regarded as substantial evidence in this context. Similar results were obtained in a trial involving adults who had been suffering from acute sinusitis for at least 8 days. The target parameter in this case was the severity score for typical sinusitis symptoms.
Patients had to have a score of more than 12 points to be included in the study. They were at least moderately, but more likely severely or even very severely affected. Then they were examined after 7, 14 and 21 days. The primary endpoint was the change in this symptom score on day 7 as compared to the placebo group. And what they found was a highly significant reduction in the mean symptom score as compared to the placebo. Another important aspect, for socioeconomic reasons as well as from the individual's own perspective, is the patient's working ability. After 1 week, 62% of patients in the EPS 7630 group were able to go back to work, as compared to only 36% in the placebo group.
The study did not limit itself to sinusitis symptoms like headaches and a stuffy nose.
In this study, they went to the extra effort of not only evaluating the symptom score, but also taking X-rays of each patient at the time of study entry and again after completion. Actually, an X-ray of the sinuses is not really necessary for a diagnosis of sinusitis. This is a clinical diagnosis based on various factors. You don't need an X-ray image for it. For the purposes of the study, however, it certainly made sense that even in an independent assessment, they were actually able to say, in all those patients that were included in the study, a maxillary sinusitis was clearly apparent in the X-rays as well, and it got better afterwards.
The studies conducted with children posed a particular challenge. For 70% of all medicinal products used in pediatrics, there are no specific pharmacological studies available. With Pelargonium Sidoides, the situation is different. In 2 placebo-controlled trials involving more than 450 children and adolescents, a very good efficacy and tolerability was demonstrated.
The Schwabe company went ahead and did placebo-controlled trials starting from the first year of life. The youngest child in these large studies, with a total of 800 children, was just barely 1 year old. And that was when it became quite obvious that EPS 7630 is very effective, that it heals infants, youngsters, as well as adolescents much, much faster. After only 3 to 5 days, they felt fitter as compared to the placebo group. We have the immunomodulatory effect. We take advantage of that to speed up recovery. But the immune system still has to deal with the pathogen, so that the memory effect of the immune system will still be developed.
But regardless of all the impressive evidence from state-of-the-art scientific studies on the mechanism of action, and the multitude of clinical trials which have substantiated the unique benefits of EPS 7630 that 30 million satisfied patients have come to appreciate, namely its quick action and good tolerability, the extract of Pelargonium Sidoides is still a subject of extensive research.
I'm positive that even more potential applications will be found in the future. After all, research is making continuous progress, so we'll be able to use EPS 7630 in all kinds of respiratory tract infections, not only in acute bronchitis, sinusitis or angina. After all, we're really only at the beginning of our research, not only with regard to EPS 7630, but with research into the mechanisms of action of herbal medicinal products in general. Today, there's a general trend towards natural and herbal medicinal products. However, the public has become much more critical. They not only want tradition, they also want current evidence, which I think to a great extent has been accomplished in the case of EPS 7630.